Hello, I am Anuradha, a first-year master's student at the CSRC. My research is to model the spatio-temporal distributions of HIV particles and nanoparticle-based preventive therapy. The human immunodeficiency virus is a virus that attacks the body's immune system. According to the WHO, there are 37 million people living with HIV in 2020. To contract this virus, it must enter the bloodstream. It is usually transmitted through sharing needles, syringes, infected bodily fluids like semen or vaginal fluids. Once this virus enters the bloodstream, it targets many cells, especially CD4 cells. These CD4 T helper cells help coordinate the immune response by stimulating other immune cells to fight infection. HIV weakens the immune system by destroying these CD4 cells. The virus is tough to treat since the mutations are so high. With no cure for this disease that attacks the human body's immune system, there is a potential development of microbicide and nanoparticles which help prevent the disease spread. Since the HIV target cells are under the epithelial cells, the virus needs to cross the acidic cervicovaginal mucus, that is the CVM layer, that acts as a barrier for the virus to move freely. The transport through the mucus allows us more time to intervene through drug-filled nanoparticles. Our collaborator, Dr. Bibo Dion, is developing drug-filled nanoparticles with a combination of tenofovir and marovirog drugs for the preventive therapy. When these nanoparticles are applied, they attach to the virus and destroy them before the virus reaches the target cells beneath the epithelial cells. Previous studies have indicated that the motion of the virus depends on the acidity, because acidity increases the adherence of the virus to mucus sites. Moreover, the acidity of CVM might change in the presence of neutral semen. Since the motion of the virus in the acidic CVM is hindered, accurate modeling is necessary that can incorporate hindrance due to adherence in acidic media. We model the spatial temporal dynamics of virus concentration using two components, diffusion and hindrance. Where diffusion is modeled using Fick's law and pH-dependent hindrance is modeled using the biochemical reactions. Here we have the virus and mucus interacting with attachment and detachment rates Kb and Kc. Vm represents the mucus with the virus attached. Here Kc is the rate at which CVM-bound HIV detaches from the CVM. We then solve the reactions to find the hindrance. This is the final model for spatial and temporal dynamics for free virus in mucus. Calculating the time taken for the virus particles to reach the target cells, we find that the transit time is 3.3 minutes when the pH of the mucus is neutral, and the transit time is 13.9 hours when the pH of the mucus is acidic, giving us about 13.5 hours to intervene through drug-filled nanoparticles. When these nanoparticles are applied, each nanoparticle attaches itself to the virus and destroys it, ensuring no virus reaches the target cells. From the graph on the right, we see that the mucoadhesion rate increases as the acidity of the mucus increases. Here is a spatio-temporal distribution of HIV particles in the mucus in 1D and 2D cases. In the second graph, we see that no particles cross the mucus within 5 minutes when the pH is acidic, that is 4. Whereas over 100 particles cross the mucus within 5 minutes when the pH of the mucus is neutral, that is 7. In the future, we are planning to model the movement of virus through the CVM with the application of the nanoparticles and their interaction with the virus. Thank you.